Hello, I am going to introduce Dr. Khokhar, who is going to tell us about the genetics of the edibles. And all the cannabis vaping and how this relates to genetics. He's done extensive research in animal models, and this is the field of translational neuropsychopharmacology. I know what neuropsychopharmacology is, but I'm a little bit up in the air about what the translational part is. So I hope that Dr. Khokhar is going to explain all that. And I know you will have a lot of questions like I will. So please put your questions in the question and answer box, and we'll keep track of those. So let's turn it right over to Dr. Khokhar.
Thank you, Dr. Kamer, and thank you to IASIC, and thank you to Drug Free America and everybody else for being here today. I'm looking forward to this conversation, and hopefully we can all learn together. The title today is Cannabis Vaping, Edibles, and Genetics, and what we can learn from animal models. And I think that's where the translational part comes in. So many of our questions around cannabis are hard to answer in humans. We can't give people cannabis and see how they turn out. And so what a lot of our findings in humans remain association-based findings. And so what we can do is reverse translate those back into animal models where a lot of the brain mechanisms that cannabis acts on are conserved and try to understand what the causal basis of a lot of the effects of cannabis are. And so before I start today, I'd like to acknowledge that the work that I'm presenting today was performed both in Guelph, Ontario, and in London, Ontario, in Canada, on the traditional lands of the Mississaugas of the Credit First Nation of the Anishinaabeg people, the Haudenosaunee, the Lenapehoik, and the Chinookan nations, on lands connected with the Dish with One Spoon Covenant Wampum. And for those that haven't heard land acknowledgement before, we do them often in Canada to remind ourselves of as neuroscientists, we try to understand, the brain up here, the most complex mysteries of this brain, but you might forget that it is done by flawed human beings, us. And sometimes neuroscience has been and continues to be used to uphold and entrench systems of racism and colonialism. This is not something we shy away from, but instead we reflect on this and try to understand how we can contribute to understanding and mitigating the long-term consequences of settler colonialism and genocide on the brain, body, and society in Canada and across the globe. So speaking of Canada, cannabis, we're no strangers to cannabis. Canada legalized cannabis now almost 8 years ago or 7.5 years ago. And so that was when I was starting the lab. And so it was high time to be doing cannabis research in Canada. There will be a liberal sprinkling of dad jokes throughout. So I'll be here all night, folks. Speaking of Canada, Canada has had one of the higher rates of cannabis use in the country across the globe. And so when you look at past 12-month use, over a quarter of Canadians report using cannabis. But especially when we look at that 16 to 19 or the 20 to 24 demographic, that's when those rates are even higher. So almost 40% of that 16 to 19-year-old, and then half of those that are between the ages of 20 and 24 use cannabis. And not only are the rates high, but it's also important to consider the perceived risk associated with cannabis use. And so this is data from Statistics Canada. And, compared to tobacco smoke, which is, the single largest preventable cause of death worldwide, but compared to that, even nicotine e-cigarettes, which are popular because of their safer profile compared to cigarettes, we see cannabis, especially in the various forms that it comes in, and even compared to alcohol, is perceived to be safer. Especially when taken via edibles. And so it's important to try to understand whether these perceptions by the Canadian public are in fact based in evidence or not. And so we'll tackle this in three different ways. We'll talk about vaping. So vaping right here, and we'll talk about edibles and what the effects of those might be, and then try to understand what the genetic basis of cannabis use are as well, and how we can use animal models to really get at what might be the causal basis of these effects. Adolescence and cannabis and talking about it is nothing new. I am not the first person to talk about it. I will definitely not be the last to talk about it. And we've seen all sorts of messaging going all the way to this is your brain on drugs. Eggs don't really have much to do with cannabis, except maybe you like eggs after you've had cannabis. To more recently, the Stoner Sloth campaign that came out of Australia, which, those that haven't seen it, is sort of like that scene from Zootopia where the sloth is at the DMV typing really slowly. And again, cannabis doesn't really slow you down. It's actually paradoxical where it might result in locomotor slowing, but because it might make you more impulsive, it ends up being a wash. And so it's important to then try to figure out what is real. What are the effects of vaping on the brain, for example, and how can we use that to better inform young people about their decisions around cannabis? And so this is recent data out of Canada, and the same as applies to the US as well. Teenagers who use cannabis or marijuana are shifting from smoking to vaping. Should we be alarmed? Well, at least we should be studying it and trying to figure out what is it that vaping does to the developing brain. Adolescence is a unique period of development. I'm just going to move this bar, or maybe I can't move it. I'll just leave it. In which the brain is going through a lot of changes, and we'll talk about it a little bit on the next slide. But what we know from human association level data and brain imaging studies is that any substance use during this period of adolescence is associated with a change in the risk trajectory for the development of three main areas or domains. Substance use disorders. So the earlier you start using a substance, the greater your risk of developing a substance use disorder, either for that substance or another substance. Psychopathology, so the relationship with serious mental illness. And then the last one being cognitive deficits, and it differs based on the drug you use. But those are the main domains where we see an effect of this in the human association study. And this is because the brain is going through a lot of changes. This is a busy slide on purpose. What it tries to capture is the main neurotransmitter systems in the brain. And you can see each one of them during that adolescent period is going through a flux or a change from the glutamatergic system, which is a primary excitatory system in the brain, the dopamine system, which is a reward related system in the brain, the nicotinic system, where nicotine acts on, the cannabinoid system, where cannabis acts on, the opioid system, where opioids act. They're all going through considerable flux during this time. And what that means is that that's the prism through which any exogenous influence, any drug from outside, cannabis, for example, is now going to have its influence. And that trajectory that we talked about on the previous slide is going to be impacted as a result by these changes that are happening on the ground. And so, how do we study this? Well, we take cannabis from the Ontario Cannabis Store. So this is cannabis that people use. And we actually look at the Yelp reviews as well to ensure that this is something that is actually used by people. And then try to use it in a form that is similar to how humans use it. Many before me have actually injected rats or mice with doses of THC or synthetic cannabinoids, but no human injects cannabis. And so it's important to mimic the routes of administration that the humans use. And again, that's where the translational part comes in, is where taking how humans use the cannabis, bringing that back into an animal model to try to see if we can assess the effects of cannabis as humans would use it. And again, because this is about adolescence, so postnatal date 28 to 42 is what is the adolescent period for a rat. And the nice thing about this is within 100 days, we can then do the entire study and really try to figure out what is happening to the brain. And then another piece where the translational piece comes in is not only are we trying to mimic exactly how humans use the cannabis, but how it's studied, we're also using MRI just like it's used in humans to be able to study the changes in the brain. But now we can attribute it to cannabis alone, because in the humans it could be other socio-demographic factors, where they live, what genetic background, etc. in this experiment, the only thing that's different between the groups is the type of cannabis they were exposed to. And again, here we're using very small amounts of cannabis, so 0.25 grams put into the volcano, which is, the OG vaporizer. It's an oldie but a goodie. Before COVID, I guess. It used to fill up a bag and then you could pass it along. In COVID, the whip attachment really took off, and that's what you can see here. And what we just do is basically put the cannabis in there, hook up the whip attachment, and then pump it into a cage where 16 rats, 4 in each compartment, are basically having a party. These are adolescent rats, and they're essentially hotboxed with the cannabis vapor that comes out of the volcano vaporizer. And then the animals are allowed to grow up into adulthood, where we first assess them for behavior, and then we assess the changes in their brains. But when it comes to vaporizing cannabis, it's not like injecting cannabis into an animal. Partly because you don't know, while you are the one that turns on the vaporizer, the animal still has volitional control. They can decide to go stick their head right where the vapor is coming out, or they can decide to go stick their head in the other corner. They can decide to hold their breath, or they can decide to take more breaths in. And so it's important to anchor it to something real. In this case, we anchor it to the concentrations of THC, CBD, and the primary metabolite of THC, 11-hydroxy-THC, in the plasma. And what you'll notice is that these are low levels of cannabis. This is not like vaping concentrates or dabs or shatter. This is flower that's vaporized, resulting in low levels of cannabis, much lower than any of the injected experiments have done in the past. And in addition to that, we also see the behavioral effects, where it affects motor activity, it affects body temperature, and it also affects tail flick latency, which is a measure of how much pain the animals can perceive, where the high THC cannabis produces the highest level. What's also important here is the labels on those products from the Ontario Cannabis Store were in fact telling the truth. So the highest THC cannabis produced the highest levels of THC. The highest CBD cannabis produced the highest levels of CBD, and then the higher THC cannabis also had the highest levels of that primary active metabolite, 11-hydroxy-THC. So that was during adolescence. Now we let the animals grow up. So basically, for another 20 days after their last exposure to cannabis, they are allowed to grow up without any more exposure to cannabis. It's kind of like, the model is, you're going to live with your auntie and uncle in Bel Air. So now you don't have any more access to those hotboxing parties anymore. And now we're assessing these outcomes in adulthood. What this is, it's a sign tracking task. It's what in humans would be closest thing would be cue reactivity. What we do is the animal is in a box with levers and a food pellet dispenser. And the lever comes out, lever goes back in, regardless of whether the animal presses the lever or not, food is delivered. So the animals begin to form this association with the lever, where it's an appetitive association, where they'll start to chew at it, gnaw at it, bite at it. The best example I can think of is, the golden arches for McDonald's have never actually given us Big Macs, but every time we drive by, we associate that with the Big Macs, and we might pull into the drive-through. And so the same way the rats form this association, where they now try to bite at the lever, even though it actually doesn't produce any reward or result in any food pellets. And what we see here is the blue is the control group, that these animals learn this just fine. The balanced group actually learns it just fine as well. But both the high THC and the high CBD groups actually fail to learn the task. And this is important because the drug is no longer on board. These are long-term effects after very low levels of THC or CBD exposure. And so this was on the appetitive front. That's not the only type of learning that we do on a daily basis. We often have to learn about what things might be aversive. Is the hot stove going to burn us? Is the scary sound outside scary and we should learn to run away from it? And so we also assess their learning on an aversive task. What this task is basically a light comes on and it predicts the appearance of a light foot shock. And I've tried the foot shock on myself, and it is a mild foot shock. And what that does is the animals can learn quickly that they if they look at the light and they run over to the other side. So there's a guillotine door right in the middle. And if they run over to the other side, they can avoid the shock altogether. And the cool thing about that is the animals learn this pretty quickly. This is one day. In 10 trials in one day, the animals learn that the control animals quickly learn that they can avoid the shock altogether, almost 80% of the time. All three types of cannabis actually prevented the animals from learning to be able to actively avoid the shock. And again, unlike the previous graph, there the balance looked like it was protected against, that deficit in appetitive learning. Here, if anything, they're worse off. And so your mileage may vary depending on the type of learning we're talking about. But just like we talked about in that previous graph, right? The three things that we talked about, the first one was early when we talked about risk trajectory. The first one was future substance use disorder. The second one was psychopathology. And the third one was cognitive deficits. And so we're seeing the cognitive deficits with these low levels of cannabis already. The next thing what we can look at is pre-pulse inhibition. It's often referred to as sensory motor gating. Pre-pulse inhibition can actually be done in humans as well. The example I like to use is if you walked into a room and I was hiding behind the door and I jumped out and I said boo, you would get startled. But in the process of jumping out to say boo, if I knocked over the garbage can would have made a sound, and now you're startled to my boo won't be as large. And that's called a pre-pulse inhibition. So if something comes before, a pulse comes before a startling sound, it inhibits the response to it. But when you look at somebody with schizophrenia, for example, or bipolar disorder, they have disruptions in this pre-pulse inhibition. And what we see is the animals in the same thing, where if they're given a pulse before the startling sound, if you look at models of schizophrenia, they will show disruptions of pre-pulse inhibition. What we see here, though, is that with low levels of cannabis exposure during adolescence, we don't see the same disruptions in pre-pulse inhibition that have previously been seen with high levels of THC exposure. And so while there were learning deficits, there isn't that sensory motor gating deficits that can be associated with psychopathology that are being seen in these animals. So next, we looked at the MRIs in these animals, and yes, this is an MRI for rats. What you can see there is the small bore through which the animals are put in, and we can do the same analyses that you would do in humans that are in the MRI. And so that's where we can look at the connectivity within the brain just like we can look at it in humans. And so in the first graph, what we look at is functional connectivity, sort of where how the brain regions are functionally jiving with each other. Think of it like dancing with a partner. If you're any good, then the two of you your movements would be in sync. And that's what you're looking at here is functional connectivity, where the use of oxygen in one brain region and the use of oxygen in another brain region are in sync. If you're not connected, or if you're not good at dancing, then you're not going to be functionally connected or coupled. And as a result, your connections might be off. And what we see is that the CBD group compared to the control group and the THC group has significantly higher functional connectivity across different brain regions that are involved in somatosensory, somatosensation. They're involved in visual cortex or visual function. They're also those that are involved in the raphe-median or the pontine reticular nucleus. And so different parts of the brain are differentially affected by CBD treatment versus either the control group, which just got hot air, or the THC group, which had the high THC CBD. In addition to that, we can also look at the highways that connect the different brain regions. And what we see with the highways is there's also changes in connectivity across different brain regions. So when we look at striatum, the ventral thalamus, the insular cortex, the endo and piriform cortex, we actually see increased connectivities or an increased number of highways between these regions in the cannabis-treated groups compared to the controls. And it's important because these regions have been implicated in addiction, for example. And then on the other side, we actually see fewer fewer highways between the cannabis exposed groups compared to the control group in regions such as the entorhinal cortex, the interpeduncular nucleus, the pontine nuclei. And these are regions involved in spatial navigation as well as sensory motor gating. And so, depending on what brain regions you're looking at, the effects are different, which is important to know because these are not brain-wide effects that affect all brain regions differently. Depending on which brain you're in, region you're in, the effects might be different. All the data that I presented so far was in male rats. When we look at female rats, we actually see that they're protected against the effects on learning, as well as on the functional connectivity and on the aversive learning. And there might be something different between the males and the females. And that's where we go back to those pharmacokinetics. I said earlier that the labels aren't lying when it comes to how much THC or CBD any of the groups had. But what's interesting is if you're a male or a female rat, the amount of THC that you get out of the same product, the high THC product, high CBD product, is very different. And that's because in these adolescent animals, the volume of distribution where the drug can get to can be different between males and females. And so it's important to not just be making sure that the same amount of drug is vaporized or given to the animals, but also making sure how do the animals receive it or absorb it, but also how do they metabolize it? And again, here we see that the females are able to convert much more of the THC into that active 11-hydroxy metabolite, giving us an important consideration around how THC might act in males versus females. And again, we see even the behavioral effects can be different, where only the high THC cannabis produced increases in that tail flick latency in the males. In the females, all three types of cannabis increase the latency, again reflecting differences between males and females. One important thing that we've been trying to figure out is why. Why do males and females differ in terms of why are maybe adolescent females protected against the effects of cannabis? And one of the places where we've seen it, and this is a busy graph, but what it basically is trying to show you is we expose the animals to the same three types of cannabis that we exposed them to in the previous experiment. We took the brains from it and looked at the helpers in the brain, the cells that are involved in repair, the cells that are involved in, they're called the microglia. They are the immune functioning cells of the brain. And what we see is that when we look at the brain of these animals, we see that the male animals had significant reductions in the number of microglia after all three types of cannabis exposure, whereas the females did not. And that might be one place where the one reason where we see, because of which we see the differences. And again, it's not just the number, but even the shape of these cells is different, where we see reductions in the fractal dimension here basically is referring to how spread out those microglia are. And again, we see a reduction in that as well. So there might be an effect on immune function. The next thing we'll talk about is cannabis withdrawal. Cannabis withdrawal for a long time wasn't thought to be real. But if even Snoop Dogg can complain of cannabis withdrawal, then maybe it's important that we look into it. And so that's what we did. We don't really have great models of assessing withdrawal in rodent models. And so we did the same thing where we took that high THC cannabis, exposed animals to it 3 times a day, every 8 hours for 7 days, and looked at whether the animals showed withdrawal. We could have just taken the cannabis away, but that's not enough. Sometimes you need to induce withdrawal in these animals, and how you can do that is by blocking the CB1 receptor. That's the receptor where THC acts, and we can see that after the animals have received THC and then get the rimonabant, which is that CB1 receptor antagonist, we see that in animals that didn't get cannabis, the rimonabant doesn't really do anything. No significant changes. But after this is after the last exposure to cannabis, we see that the animals are traveling less and showing decreases in body temperature, showing that the cannabis works. But when we give them the rimonabant, they start to show these symptoms of withdrawal, where they are increasing their blinking, they're shaking their bodies more, their head and body shakes more, they have greater tremors, they're writhing. It's a whole scale of somatic signs of withdrawal that we can see. We also looked at whether they needed the rimonabant, the CB1 antagonist, to show withdrawal, or even whether it would appear spontaneously. And even in the animals that did not get rimonabant, was just given a saline injection, we see that there is an increase in the total withdrawal score compared to those control animals, suggesting that even without the induction or of the withdrawal, the animals still show withdrawal. And now we think we have a pretty good model of withdrawal where we can test out different drugs with the hopes of reducing withdrawal symptoms, which remain one of the biggest reasons for people relapsing back to cannabis use. Now we get to the edibles. You know, when I was a kid, people used to warn about razor blades in your Halloween candy. And now it's cannabis edibles, right? And at least in Canada, the cannabis edibles are so expensive that I hope nobody's giving them to the kids. But it is still important to study because recent years we've seen increases in pediatric hospitalizations caused by cannabis. And in Canada, it's important because we kind of did an experiment where every other province, which is kind of like a state, legalized all types of edibles, except for Quebec, which is the primary French-speaking province in the country. And they said, we are not going to legalize any types of cannabis edibles that could be remotely appealing to kids. And so they only allowed like dates infused with cannabis oil or rutabaga flavored edibles, and nothing that would look like candy. And, this is important because clearly public policy matters, because compared to that province that did not legalize all types of edibles, all the other provinces had double the cases of poisoning. And it's not just a Canadian phenomenon. As we can see in the US, the number of young children who accidentally ate cannabis edibles jumped 1,375 percent in the 5 years leading up to 2023. And so it's happening across the board. In Canada, the teen use of cannabis edibles has also increased 43% after legalization. So again, making it important to study. So how did we do this? Again, going to that translational neuropsychopharmacology piece, it was important that we do exactly what humans do. And so Zamnesia has this recipe for cannabis-infused Nutella. So we did exactly that. We took some Nutella, and rats usually love Nutella, and we mixed in some cannabis oil, again, cannabis oil that was available from the Ontario Cannabis Store, and has 31 milligrams per gram of THC. We mixed it into the Nutella and let the rats eat it. We assessed their brain activity by electrodes that were implanted into their brain, but we also looked at behavioral effects as well as the brain and blood concentrations of THC. So what we found is, again, just like we showed before, there are considerable sex differences in how much THC is found in the plasma and the brain. So we looked at THC and that active metabolite, and not only do females have higher levels of THC, even though the same dose was given, they also have much higher levels of that 11-hydroxy THC compared to the males. They also have much higher levels of brain THC and 11-hydroxy THC. And again, you can see here that compared to the vaporized route, this graph you've seen before, the concentrations both in the plasma, but especially of the 11-hydroxy THC, are much greater than what we saw before with the vaporized THC. Again, it's because it's through the edible route, so it goes through your stomach, gets to the liver, a lot of it gets metabolized, so you end up getting a lot of that 11-hydroxy THC, which is just as active, if not more active, compared to THC. And so this is why you see the effects of edibles lasting for so much longer than regular forms of vaped or smoked forms of THC. We also see that the body temperature reductions last for up to 24 hours. The changes in tail flick latency last up to 24 hours. And even the distance moved also those reductions last up to 24 hours. When we look at the brain, we also see long-lasting changes across the brain, both in terms of the electrophysiological signal, where there's these massive reductions in the prefrontal cortex, the nucleus accumbens, the cingulate, the hippocampus, in what would be called gamma power, which is the frequency band that has previously been associated with schizophrenia, for example, and we see similar reductions after a single exposure to these high doses of cannabis edibles. And so why we wanted to do this is partly because if we can create a model, then maybe we can use that model to create drugs that would be able to reverse these poisonings in these animals. We didn't stop at just with adult animals, as the cannabis poisonings are rising mostly amongst kids. We also tried a model where we used a juvenile animal, and so the exposure happened on postnatal day 35. And again, what we see is that now the effects don't just last for 24 hours. They may last up to 14 days, depending on which measure. So body temperature comes back by 48 to 72 hours, but the distance moved doesn't for 120 or even up to 144 hours, and then it starts to come back. And then the tail flick latency, that anti-nociception, actually remains high for up to 14 days. Again, suggesting that in a juvenile animal, these effects may last much longer. What's even more interesting is that one exposure to the cannabis edible, if you let the animals grow up into adulthood and now subsequently expose them to cannabis vapor, their response to cannabis is very different. Here you can see that compared to baseline, they're all in the animals that were not exposed to cannabis, as well as the females, which are here. But here, what you can see, that animals that were not exposed to the cannabis edible during adolescence, or at that juvenile stage, show reductions in motor activity after cannabis exposure. Whereas the animals that were previously exposed to cannabis just that once actually now don't show those same reductions in locomotor activity. And in fact, on the tail flick latency, which they already had higher tail flick latency before they ever at baseline, when you expose them to cannabis again, they show even higher tail flick latency. So it's really changing even how they're subsequently responding to the cannabis. And so it's important to not just be creating this model, but also recognize the long-term consequences of such poisoning cases. And hopefully in continuing ongoing studies, we can do something to reverse some of this as well. Now getting to genetics. Genetics is complex, but if a company on Instagram that tries to sell consumer genetic products can bring it down to one gene affecting whether or not you love weed, it's probably important that we look into whether this claim is true or not. And it is based on science. I'm not saying that they're not based on science. The largest genome-wide association studies of lifetime cannabis use, which isn't the most specific measure. It's basically asking whether you have or you haven't used cannabis ever in your lifetime. One is the same as 100 times. But when we look at the genetics of that, we see that CADM2, which is a cell adhesion molecule 2, shows up as a significant genetic hit for cannabis use in a genome-wide association study. Recently, we've actually also looked at it in the context of cannabis use frequency, so how often you use cannabis. And again, the only significant hit that we see is CADM2. It's a synaptic adhesion molecule. It's kind of the glue that keeps the neurons connecting to each other. It has previously been associated with other drug use like alcohol and nicotine use as well, but as well as cognition, psychopathology, metabolism, and neurotransmission. And so it's important that we try to figure out, what does this gene do? So what we did is took a mouse model where we knocked out the CADM2 gene. So now this animal does not express the CADM2 gene at all. And what we can do is, again, taking cannabis oil from the Ontario Cannabis Store or pure THC, we can see and let the animal tell us whether it prefers cannabis or it doesn't prefer cannabis. In humans, all of the variants of CADM2 that result in greater risk of cannabis use result in greater activity of CADM2. In this case, we're removing the activity, trying to see what it does to their cannabis use. And so again, with edibles, we see that the knockout animals, so the animals that did not have CADM2 in them, they prefer cannabis or THC containing cookie dough much less compared to the wild type animals or the heterozygous animals, which have the half as much of the gene compared to the wild types. In addition to that, even with cannabis oil, now with cannabis oil containing cookie dough, we see the same thing. The knockouts prefer the cannabis containing cookie dough much less compared to the controls. And even those that had that half as much expression, as the dose of THC increases, they start to prefer cannabis less and less. What's also interesting is these mice, when it comes to the cannabis oil containing cookie dough, the females prefer it much more than the males do, which is important because in humans, while most types of cannabis use are male dominant, the only one that's not is the use of oral or edible types of cannabis. So again, the mice are maybe not such a bad model for studying these uniquely human behaviors. And so what else did we do? Why are these animals showing less of a preference for cannabis? Could it be that they're either absorbing it differently or does it hit them differently? And how you can assess that is what's called a cannabis tetrad. So these are characteristic behaviors that are seen after any time a drug that acts on the cannabinoid system in the brain is given to an animal. And so it results in decreased locomotion, decreases in body temperature, decreased motor rigidity, as you can see here with catalepsy, and then increases in antinociception, increases in how much pain they perceive. And so we see at baseline, the animals have some differences. You know, these animals, the knockout animals already move more than the wild type animals. They also show some body temperature differences and even in latency to show pain. When did we give them THC? And we gave them 2 different doses of THC. 3 mg per kg, that's a dose that doesn't do much in normal animals, as well as a 10 mg per kg dose of THC that actually hits them. And you can see that the 10 mg per kg dose of THC results in body temperature decreases, whereas the 3 mg per kg dose doesn't. And after 10 days of exposure, that effect goes away because the animals have now built tolerance to the effects of THC. When we look at that latency to show pain on a hot plate, we see that while the low dose did not do anything in the control animals or those half-expressing animals, the knockout animals now actually show greater antinociceptive effects or pain-relieving effects of THC compared to the controls. And then at the higher dose, all animals showed the same levels of antinociception. This is where it gets really interesting. When you think of somebody using cannabis, you're associating it with them moving less. And so we see that a 3 mg per kg dose doesn't really do anything, but a 10 mg per kg dose significantly reduces locomotion in the control animals and in those half-expressing animals. But whereas the animals that don't have any of the CADM2 gene, they actually show they were already moving more at baseline. When they're given cannabis, they actually move even more. And then at that 10 mg per kg dose, where all the other animals are not moving or are significantly reduced their movement, the 10 mg per kg dose, the knockout animals are also again moving, suggesting that this gene really does impact how the cannabis hits these animals. Now, going back to what I showed earlier with that sensory motor gaining, because this gene has been implicated in psychopathology, and we see a lot of overlap between schizophrenia and cannabis use, for example. We wanted to see whether the genetic risk for cannabis use or the genetic risk for something that changes cannabis use would also affect psychopathology or sensory motor gating, as we talked about earlier. And we see that the knockout animals again showed disrupted pre-pulse inhibition, just like we were talking about earlier, showing that not only does this gene affect cannabis response, but it might also affect the risk for psychopathology. Since even the 3 mg per kg dose of THC was making those animals move more, we used an even lower dose of THC to try to see how THC affects it. And again, we see that while the knockout animals still remain low with their pre-pulse inhibition, now those heterozygous animals actually are no longer different from the knockout animals, suggesting that maybe as THC comes on board, even those half-expressing animals now start to act more like the animals that are knockouts or where the gene has been deleted. The other piece of it is it just like the males and females we talked about earlier, are they just different in how much THC gets into the body? And no, that's where we don't see differences. We don't see any differences between the genotypes. We still see the differences between the sexes, but we don't see any genotype related differences, suggesting that this is directly due to how their brain receives the THC or acts on the THC compared to their bodies or how it's absorbed or distributed across the brain. The other thing we did, just like before, we looked at the connectivity of their brains. And this is where it gets really interesting. While there were no differences between the half-expressing animals and the knockout animals, or the wild type and the half-expressing animals, between the wild types and the knockouts, we see significant differences in that the blue color there is the midbrain. The midbrain connectivity with a lot of cortical and other brain regions. And what's really cool is in human studies, with people with cannabis use disorder, people with cannabis use disorder have been shown to have greater connectivity of the midbrain with other parts of the brain. These animals that prefer cannabis less actually now have lower connectivity of the midbrain with other brain regions. Again, emphasizing that animal models can prove valuable in trying to get at the causal basis of either genetic associations or the brain imaging associations that are seen in humans. So that actually brings me to the last slide. This is my future directions. We've actually been now have rats that are nose poking to receive cannabis vapor. We also have done a couple of studies in marmosets. And what's really cool about marmosets beyond their more developed brain is they have two twins multiple times a year. And so you can expose one to cannabis and not the other and maintain the same rearing environment and really assess the effects of cannabis on that developing brain. In addition to that, we've actually also been looking at whether blocking the CB1 receptor or changing how THC is metabolized can help reduce the impact of those cannabis poisoning in that model with the Nutella that we talked about earlier. And so hopefully the next time I get to speak to you, I'll be able to present some of those findings. And with that, I'd like to thank all the folks in the lab that all do all of this work. So Haley Thorpe, Hakan, Carling, Jaden Smith, Patrick McCunn, who've done a lot of the work that we presented here, as well as my funding sources and my collaborators. And I'd love to answer any questions that you have.
All right, thank you very much. Certainly covered a lot of territory. It's a lot of territory. So I'm going to go through some of the questions that we had in the question and answer box. So the first question from